Okay, I must say it is both an honor and a pleasure to introduce Carl June. So I've known Carl for over 30 years. I've followed his career. He's done great science through the years and now has really, I think, made one of the most spectacular developments in all of cancer. So just, you see here, Carl is an endowed professor of immunology in the Department of Pathology and Laboratory Medicine at Penn. He's the director for the Center of Cellular Therapies. Interestingly, and you always knew that Carl was both dedicated and focused because he is a graduate of the Naval Academy at Annapolis. From there, went on to Baylor Medical School, went then to Geneva, where he worked at the WHO. It began to develop some of the knowledge and tools that he's really utilized subsequently. In those days, he was studying immunology and malaria at the time. And then interesting, went for further training to the Hutchinson Cancer Center and worked with one of the great men of medicine, Don Thomas. I think some of you know that Don Thomas won the Nobel Prize for really developing bone marrow transplantation. So Carl is, in fact, trained clinically. He did bone marrow transplantation and oncology there. I believe he even had boards, but in his research with Don Thomas and John Hansen and others, he actually began to realize that in bone marrow transplant, there's a very important phenomenon that occurs, and it's called graft versus leukemia. So when you're doing a bone marrow transplant, what you're doing is you're giving the stem cells of another individual, and one of the problems, you bring in T cells from that individual, they can cause what's called graft versus host disease. But what they discovered is also those T cells could kill the leukemia in the patients, and that was a very innovative observation, and it really is one of the important components of bone marrow transplant. I think, at least as I understand it, Carl began to really focus on the T cell at that point. Subsequently, he spent a lot of his focus on HIV, which in fact became a dreaded infection back in the early 80s, developed T cell therapies. I think some of the technologies that he helped develop, he's utilized subsequently. That is, in fact, expanding T cells ex vivo, using viral transfers to develop treatments. And then probably in about 2000, he turned to cancer. I understand that also is from personal reasons, and began to develop what is now the spectacular therapy, cellular therapy, what has been defined as CAR T cells. And I'll obviously let him tell you about it, but he really at that point put together both the knowledge and the technologies that he had learned before, because in this case you need gene transfer. You have to make, in fact, what are called chimeric antigens, to make this work. Again, I will leave it at that. It got breakthrough status in 2014, and just a few months ago, in fact, it was accepted by the FDA as a new drug. And just a little aside, this was done together with Steve Grupp, who Steve is one of the pediatric oncologists working with Carl. And I was at a meeting in Stanford a couple of months ago, about 2 days after the FDA approved CAR T cells, and Steve pointed out to the audience that when I was chairman of pediatric oncology at the Dana-Farber, he applied for a position there, and I turned him down. And showing my great mentoring skills, I said he might want to go to another area that's less competitive. So when I give you advice, think twice. Carl has won many, many awards. You know, he's in the National Academy of Medicine at this point. He's in the Boston Society of Science and Arts. He's won numerous awards, published over 400 papers. I think he's one of the great men of this field. I'll let you tell his story, and again, I'm really privileged and pleased to introduce him. Carl.
Well, thanks, Steve, for that great introduction. I think you've built an amazing immunology program since coming here. I looked through the faculty this morning, and I have to say, within the very competitive environment here in Manhattan, you guys win. So congratulations. Now, and you know, as a fellow, in the early days of bone marrow transplantation, the major issue was we would do an MHC match transplant. And Steve brought back some flashbacks I had from back. Those days we had immunosuppression with methotrexate and cyclosporin, and we had, you know, oftentimes 25 or 30% of the patients would die from graft versus host disease. And I watched people get a bone marrow transplant who had leukemia, and within 2 or 3 weeks, you know, a 1 kilogram liver could be turned into mush by T cells. I mean, it showed, and you could do the calculations. We infused generally around 100 million T cells, and they could kill 10 to the 12th liver cells. It's amazing. So that was an issue of, you know, activated T cells have unrestrained activity, and the issue has been, you know, could it ever be done in an autologous fashion and obviate the need for allogeneic effects? So, and that's in part what the CAR story is about. And I have relations with Novartis and Immunity today. So we're at this time now in oncology where we've had, you know, for over a half a century, major therapies, but basically for metastatic cancer, they have usually failed. We're at this revolution now that, you know, many of you here are working in, where immunotherapy now, combined with other precision therapies now, is really changing the ability to actually cure metastatic cancer for the first time. And I think this is really an exciting time of how this can happen. And I'll talk about some of the issues with that. Probably the most famous slide that you'll see if you go to various immuno-oncology meetings these days is this from Ira Mellman, who made the so-called cancer immunity wheel. And you know, there are 7 steps in here on how to train the immune system to actually have an anti-tumor effect. And one of these here is step 6, which can be T cells. But I'll be focusing on that really. It's an orchestra of the innate immune system with macrophages, NK cells, T cells, and B cells. There are many different effects, and this can help orient some of those issues. So, but that wheel really was about the natural immune system. Synthetic biology then gives you a whole nother direction that's orthogonal to that, and that's the ability to enhance immune cell function above what, you know, Darwinian evolution did. So you can now make T cells not only redirect their specificity, but as I'll show, change their metabolic properties. And I think with genetic editing and as facile techniques such as CRISPR editing, we're going to see many, many issues that will make T cells both safer and more potent. And I wrote this review with Wendell Lim in Cell earlier this year, which talks about different opportunities there. So, and probably the first aspect of synthetic biology to enter medicine now has been CAR T cells. And they are a bispecific cell. Natural T cells are MHC restricted. They need an HLA molecule, and it has to be the one that the T cell grew up in with the thymus. CAR T cells are MHC independent. The T cells retain their TCR. The first CAR was a very simple one that had the zeta chain of the T cell receptor. And an amazing aspect about this is it was made for a basic science experiment with no thought of translation. It was made by Art Weiss and colleagues at UCSF in the early 90s, and he was asking how the T cell receptor worked, which was a mystery back then. And he found that this zeta chain, which ended up activating the RAS pathway, could phenocopy most of what the 8-chain, multi-chain receptor of the T cell receptor does. And later, it was recognized that this fusion protein of CD4 or CD8, the CD4 molecule could bind with high affinity HIV. And we then, with cell genesis in the 90s, did the first CAR T cell trial with this so-called first generation CAR having a single signaling domain in patients with HIV. And it had antiviral effects in a randomized phase 2 trial, but it didn't cure patients. And those studies were published in 2000. and then abandoned because the protease inhibitors got so good in HIV and we no longer had end-stage H patients, which we had been dealing with in the early 90s. But these first-generation CARs, and Zeligesher made antibody-based recognition domains then to tumor antigens, didn't work in cancer at all. They didn't survive for more than a week in the patients, whereas we showed and published here over a decade survival of the CD4 zeta CARs in patients with HIV who are on antiretroviral medications. The half-life of this CAR in a patient is more than 10 years. And so that, this is where the term having a living drug comes in. A single infusion in these patients has resulted in more than a decade of cells persisting at about a 1% level. And the patients were given no chemotherapy, and it's been safe. In cancer patients, these cells survive for about a week, so the tumor microenvironment is much more immunosuppressive, and there's many reasons for that as to why they don't survive. So what's been developed and will be FDA approved is so-called second-generation CARs, where they have either a CD28 signaling domain or 4-1BB, and I'll be talking about these in more detail. And then there are many others who are being developed, and I'll just say a little bit about the gene Icahn. But there are some even fourth-generation CARs now being tested in preclinical studies. And Icahn is a signaling molecule that arose as a gene duplication from the CD28 molecule, so it's synthetic on human chromosome 2, but it has different functions. It activates cells, but has some different signaling capabilities than the CD28 molecule. So Sonia Gaydon in my lab has made a variety of second and third generation CARs now, combining and testing and comparing 4-1BB to Icahn and in either pairwise or in triplicate. And she's done this testing the function in both CD4 and CD8 cells. And the optimal signaling domain actually depends on whether you're a CD4 cell or a CD8 cell, or what subset of cells you are. So the signaling domain that works best in a Th1 cell is different from a Th17 cell. So it's going to get much more complicated. And she's testing these in immunodeficient NSG mice, in this case with lung cancer and non-small cell lung cancer, or with pancreatic cancer cells, and looking at tumor survival in flank tumor models. And the first thing she's found is that the cytosolic signaling domain has a major impact on the cytokines made by the cell when it sees a surrogate antigen. So in this case, this is a mesothelin CAR, and the target is mesothelin, and then that triggers cytokine, which in the case of IL-2 is the winner is, if you will, is CD28, which mimics its endogenous function. And Icahn makes very little IL-2 as a CAR. But in contrast, IL-17, which is a dominant cytokine, IL-17A, made by Th17 cells and found in autoimmunity, the winner is Icahn. And interferon gamma is made by all of them. And so you can shape the cytokine profile depending on the signaling domain. Potentially that could change the propensity for cytokine release syndrome, which has been a major problem, especially with CD28 signaling. And so she then looked at the anti-tumor effects, and we have this trial, which is, it has 4-1BB and zeta. It's the same as what is just recently FDA approved, but this is against mesothelin in a pancreatic cancer flank tumor model. These are very large tumors, much larger than you see in most studies. And if you just treat with untransduced T cells, the mice die within about 35 days, or have to reach an endpoint where they have to be sacrificed. This is a trial that we have in clinical trials, a CAR, anti-mesothelin with 4-1BB and zeta, and it causes stable disease in these mice, but we also see in humans some responses with that CAR. This is the waterfall plot of that compared to the mice that get either untreated or just saline. But the 4-1BB Icahn triple CAR here, cures these mice, as you can see. So we're not sure of all the mechanisms yet. In part, it's resistance to exhaustion of the CAR T cells, and it's also enhanced persistence. So in leukemia, what correlates most with long-term survival is the CAR persistence in the patients after infusion. And this is 21 days, so from this time point here in these mice, they still have antigen, and there's much more survival of these CARs here than the CARs that have different signaling domains. So we think that that's the best biomarker we have right now to look for improved function. There are distinct gene signatures by these CARs. So if you look at principal component analysis and 3 healthy donors, there's 15 genes different between 4-1BB and, between the CD28-based CAR and ICOS, and much more distance between the 4-1BB-based CAR, which activates TRAF2 signaling pathways. These are some of the genes that are very different. Of course, when we confirm by PCR, IL-17 is only induced by the IL-17 CAR. And there's one gene that is not known to have function in T cells, but we're just looking at, it's called neuronal calcium signaling 1. And it is induced only again by the ICOS-based CAR. This is Dan Littman's model of how Th17 cells work as far as transcriptional networks, with green lines indicating activating. So BATF, for instance, activating here in HIF1 alpha, and we think this NCS1 molecule, KLF3, for instance, turning things off. So this is may offer new targets to enhance CAR proliferation. And what I'm going to do now is shift to the CAR that we're using in clinical trials beginning in 2010. It was a CD19 CAR with a 4-BB zeta. And we first began treating patients with ALL, which I'll mention. David Porter did that in first infusion here. The first patient is now 7 years out and leukemia-free. So we've had very long-term remissions there. And then in 2012, we began treating adult and pediatric patients with leukemia, as Steve Burakoff mentioned, with Steve Grubb at Children's Hospital. All of these diseases have been treated and they have all responded, and many centers have now seen this. So all B-cell malignancies have responded to CAR therapy. And the drug is the same, so in this case it's autologous T cells genetically modified. We grow them for 10 days in the lab, use a lentiviral vector to introduce this CAR. It has a constitutive promoter with EF1 alpha, and then the cells are frozen at the end of a 10-day culture period and into infusion-ready DMSO. Then just as in a bone marrow transplant, the patient's in an infusion room, the cells are brought and thawed in a 37-degree water bath and just given in a 5-minute infusion to the patients. So it's given as a single infusion. And the British have now looked at this, how is the survival compared to other therapies? What they did was the previous FDA-approved therapy for relapsed ALL, so this is twice relapsed and more. Half of our patients have already relapsed after an allotransplant, and there's never been a salvage therapy for patients who relapse after transplant. And the British pooled the published data from Penn Memorial and the Fred Hutch, and this was a survival in the published studies. It's actually better now, but that's published. And this is an FDA-approved drug. The previously approved drug to our CAR is clofarabine, and you can see it looks like metastatic pancreatic cancer, and that drug was better than the standard of care. So it's an 18-month survival, 10% survival rate. So ALL that has relapse is an awful disease in children and adults. Quite amazingly, Novartis was able then to take our single center studies and globalize it. They have now a manufacturing plant in Morris Plains, New Jersey, and they conducted an international trial, a phase 2 trial called ELIANA for relapsed leukemia ages 3 to 25. And at these centers here in the US and overseas, they now have a manufacturing plant in Leipzig, Germany as well. But this involved very complex logistics that I never thought would be possible to globalize. They requires transoceanic shipment, for all these patients treated. And amazingly, they got the same results we got in a single center study in the US, 80 to 90% CR rate, and not a single fatality. there was a 48% incidence of cytokine release syndrome, but it was all manageable with tocilizumab, which is an anti-IL-6 receptor therapy, which I'll mention. So it was FDA approved on August 30th, which was quite an amazing day for me, I have to say. We never thought that a gene therapy would be FDA approved in the US. It's been a long road. The FDA actually had a press release calling it a historic action. And so Scott Gottlieb and our administration has done something good. So these were the news headlines. But at the same time, this issue has come up, and it's a really interesting issue of a disrupting medical therapy. So Forbes published the same day the price that Novartis announced after negotiation with CMS for the CAR therapy. You know, it's an ultra-personalized therapy made one by one. And the CEO of Novartis was criticized from both sides as saying it was too high a price or too low a price. It was 36% below what Wall Street expected. And I can tell you, I mean, their profit margin on this is much less than standard drugs because the cost of making this is much higher than the normal ratio of a selling price for drugs. So this is an evolving issue, but the British looked at what is it actually worth based on the life years saved. And the British and health economists do these with QALYs, quality adjusted life years. And what they said was it was worth in this patient group 670,000. So Novartis priced it under what the British predicted it would be worth. But for the first time also, Novartis made a so-called pay-for-performance guarantee. So the hospital and the patient only pay for the drug if it works. So if the patient doesn't go into remission, it's free. That's a really big change from how standard medications have been given, where if they fail even, you know, we give reimbursement or we pay for failure. So this is a major issue now. How do you pay for expensive drugs? But if they're life-saving, what are they worth? It's a very large issue. The answer is different in the US than it is in single-payer health systems outside of the US, where it's simpler to integrate something like this. So it's an ongoing issue. Yesterday, there was an article in the new medical journal called USA Today, and it brought out a major issue, which is, you know, we've had other therapies that have happened, like bone marrow transplants, and there's no FDA approval of it, but it's paid for every day based on negotiated settlements with different healthcare systems. And what the USA Today brought out yesterday was the Novartis price there that they will pay pays for the CAR manufacturing, and it pays for tocilizumab, which will manage, and it's co-labeled with Roche, which manages cytokine release syndrome. But it doesn't pay, for instance, if you have to go to the ICU. So the hospital has to find a way to make up that revenue. And our healthcare system needs to find out how to handle that. So this is the package insert for the CAR. It's called Kymriah, which we used to call CTL19. But it has two black box warnings. One's for cytokine release syndrome, which now is a manageable, if you're trained, it's manageable, with algorithms including IL-6 receptor targeting. And in addition, it has neurologic toxicities in about a third of the patients. And that's a that's a class effect because blinatumomab, which is a CD19 bispecific antibody, also has this. The basis for the neurologic side effects are unknown. They are reversible in the case of this CAR. The cytokine release syndrome is predictable. It's the chances of having CRS, it was 48% of patients in the international trial. The chances of having CRS are based on how much your tumor burden is at baseline. So if you treat patients who have low tumor burden, they have a low probability of CRS. But high probability, if they have a high tumor burden, more than half the patients require ICU therapy or tocilizumab. The other aspect of this is this can cause now a false positive HIV assay in the patients because in the vector backbone, there are sequences derived from HIV that are not in the transgene, but so patients need to be told ahead of time that they'll have a false positive assay. As long as the CARs are engrafted, they have a signature of the HIV encounter, even though it's not that, it's a lentivirus. So how do you make T cells better? This is a review from Greg Delgoff, and we've heard a lot about checkpoint therapies, and molecules such as CTLA-4, PD-1 have been FDA approved now since 2011, and have had massive importance now in making untreatable diseases like metastatic lung cancer treatable in a subset of patients now. But the other issue, and one that I think that synthetic biology is going to be able to address in a better way, is metabolic checkpoints such as tumor making low glucose, hypoxia, acidemia through lactate, et cetera. And those can be very immunotoxic to T cells. And even tumors among themselves have different metabolism. Some, such as lung cancer, do more oxidative fatty acid oxidation, and others are entirely Warburg and do a lot of sugar burning. So the ideal T cell depends on what the tumor is, really. And so Amkar Kowalekar, my lab, has looked at this and made CAR. This is the one that's FDA approved now, CD19 with 4-1BB zeta, and compared it to the CD28 signaling domain, keeping the antibody constant. And when you add leukemia cells to these CAR cells, they triple in volume, the CAR cells do, and they kill the leukemia cells. And then they decay over time at the same rate in their volume as they go towards resting, irrespective of the signaling domain. But if you look at oxygen consumption, and this is in a seahorse assay, oxygen consumption rate, there are massive differences. They're not there at baseline, but 7 days after seeing their antigen, you can see that the 4-1BB-based CAR has much higher maximal oxygen consumption, and that persists for as long as we followed the cells. So single contact with the antigen and with this CAR, they now look very different for weeks later. And that may be important because Erica Pierce and others have shown that CARs that have oxygen burning capacity and lots of mitochondrial biogenesis, which this CAR does, those are the ones that live as central memory cells. And cells that have lots of glycolysis and are, in general, become more effector cells and short-lived. So you can now change not just the specificity of T cells, but you can change their metabolic fates. And I think that probably what we could do is phenocopy the natural immune system, where you have a nice early effector response. And then when antigen load goes down, you want to have cells persist as memory cells for immunosurveillance, predicting that maybe the best would be a mixture of these two kinds of CAR cells. That has not yet been done, but we intended to test that would be better than either one of these, supplying cells that are both very early effector cells and then cells that are more likely to stay as memory cells. In our first patients treated, David Porter had the phase 1 trial with refractory CLL, had 14 patients. We now had an opportunity to look back at them, and this is for the first year after treatment, looking at the CARs here by a PCR assay. So this is looking at integrating CARs and genomic DNA, peripheral blood. And he arrayed them in from CRs to partial responses to non-responses. The overall response rate in CLL is 57%, and the CR rate's about 35%, so it's really different than ALL, where it's 90%. But the responding patients share two characteristics. They have higher peak CAR levels than these non-responders, and they all have what we call memory CAR cells, so we can look a year or 5 years later, and they still have functioning CAR cells, and the non-responders don't. We don't know all the reasons that separates this differential engraftment and persistence of the CAR cells. We think it's both some cell intrinsic issues in the T cells, namely exhaustion, and also the tumor microenvironment, which may be more immunotoxic in some patients with CLL than in others. But the last patient that we had here had the latest peak, and we've now been looking at him, and we looked in detail at the PK of his CAR cells, and there were 50 patients that we had to compare who got the same CAR. So these are adults given infusions, again, looking on a log scale. When you get up to here, about more than 50% of your circulating cells are CAR cells. That lasts for maybe a week to 2 weeks, and then the cells decay and go down at a 2-year time point to low levels. We can show they're still functioning, but they're at low levels, less than 1%, in both bone marrow and blood. Patient 10 is delayed, as you can see in the red, but he had the same decay rate. And so we've looked into that. He was an interesting 77-year-old man with very high levels of leukemia in the blood and large nodal masses. This is peri-aortic. He had also mediastinal masses and a baseline. And a month later, which was an initial endpoint in the study, he still had, on a bone marrow biopsy, packed bone marrow, and he still had leukemia in the blood. But then he had tumor lysis syndrome, that began day 50 after treatment. He required the same therapy as the rest of our patients, and then has entered into a CR. He's now 4 years out in a deep remission. And we've looked now in detail at what happened in him, why he had this delayed response. The response was comprised of CD8 cells, and it was a single TCR, because we sorted the cells. We could use an anti-idiotype, which binds to the CD19 CAR. Initially, this is in gray, is low level, low abundance insertions. There were over 1,000 different insertions in what we infused into this patient, and they were a mixture of CD4 cells and CD8 cells. But on day 120 or 121, they're all basically, there's no gray anymore of low abundance insertions. They're all the gene down here, if you can see the color, TET2, is a single integration site, a single TCR. And we then just sorted, this is bulk cells in his blood, and then looking at integration analysis done with Rick Bushman at Penn. And the CD8 cells showing the same answer. So he had a polyclonal set of CAR cells, which is what we always see in patients, but he evolved into being a monoclonal population at the time of heavy lifting when tumor lysis syndrome happened. He's an outlier in that respect. The rest of the patients look like this throughout. They don't have dominant integration sites. So what happened? So he had a single copy of the CAR integrated in chromosome 24, Q24, and it turns out it was integrated into the TET2 locus intronically between exons 9 and 10. And what this did was we found 3 different transcripts that created premature stop codons. So it's at least a 1 allele knockout of TET2, because the business end of TET2, which is dioxygenase, requiring 2-oxyglutarate and iron as a cofactor to demethylate methylcytosine on DNA, was now no longer expressed in these cells. So he's hypomorphic at TET2. And TET proteins really are a very interesting class of, they modulate epigenetically DNA methylcytosine. There are 3 of them in human T cells. And TET2 as loss of function can be the first mutation in AML. And the group at Stanford's really shown a lot of that. But it also increases stemness, and that's work from Peggy Goodell and others at Baylor. But it can be mutated in, for instance, T-cell angioimmunoblastic lymphomas and so on. So it has effects on stem cells, but a mutation in TET2 alone is not sufficient for malignancy. And most patients who have CHIP, which is clonal hematopoietic hematopoiesis, so maybe 10% of 70-year-olds and above, they have loss of function mutations in TET2. So it gives a stem cell gain of function. So we learned a lot out of this patient 10. Oftentimes we're asked, what's the minimal dose of CAR T cells that you need? Because it's a replicating cell. The answer is you need a dose that will give engraftment of the infused CAR T cells and then subsequent proliferation. In patient 10, it was 1 cell. So the clonal progeny of 1 cell cured that patient. Because they're fate mapped, we can show that. At the time of tumor lysis, it was all the progeny of a single cell that was a rare cell on what we initially infused in him. Now, he's 4 years out now, and the CAR T cells are now at low abundance in the patients. And we don't know if this would be safe to do intentionally on patients, but it does show if we could do this reproducibly, the cost of goods of manufacturing CAR T cells would go down exponentially. And instead of infusing 10 to the 9th CAR T cells, we could infuse maybe 100,000. So that's where the field will go. It's going to require engineering, and we have the tools to make cells have high proliferative capacity. So what I'll turn now to is some issues in solid cancers. We're going to see many different indications with CAR T cells in bone marrow malignancies. I think basically all bone marrow malignancies will have approved either T cell engineered or T cell receptor or CAR T cell engineered for them over the next 5 years. But solid tumors are still a much more challenging issue for a lot of reasons. One thing we've done is ask, if you have solid tumors in vitro, and we've done this in either two-dimensional plastic cultures or organoid cultures, can an engineered CAR T cell actually kill them? And we've taken primary cancer cells from our spore programs in ovarian cancer, lung cancer, and pancreatic cancer, and made CAR T cells to them, and used CAR T cells that either have an irrelevant receptor such as CD19, or which then controls for allogeneic effects, or mesothelin, which has, which is on these solid tumors, and ask, can they kill? And this is BXPC3, which is a ATCC pancreatic cancer cell line, and there's specific killing that the non-specific CD19 CAR does not kill. If you look 5 days later at a mixed culture where we added tumor cells and CAR T cells, there's no live tumor cells left here, but the allogeneic T cells can't kill. So we haven't really known that in the past, because when people did allogeneic effects, you're really dependent on the affinity of the allogeneic T cell receptors for the tumor cells, and many tumor cells downregulate class 1. So we couldn't really see this. With a CAR T cell, we have a high affinity receptor, and it doesn't care if the tumor cell is expressing MHC class 1. So they can kill. The issue is there's a toxic microenvironment, there's trafficking limitations, and there are checkpoints. So this is another example of this, another pancreatic cancer cell line, and using the Excelligence assay. And there, it's measuring capacitance. If the tumor cells are growing, they just make more capacitance in the culture bottom. And allogeneic T cells, or the CD19 CAR cells, the tumor cells grow just as well as the cells growing in media alone. But if we add an anti-mesothelin CAR, it kills almost as well as Triton detergent does. So CAR T cells can kill if they get there, and I think that's a good news issue. The bad news is we have a lot of checkpoints to solve metabolic issues, trafficking issues, and so on. But I think that's what the field can do to solve that. We actually did the first CAR T cell trial in humans back in the 90s with Cell Genesis, but it wasn't published until earlier this year. We didn't bother publishing it because the results were so bad. And so we decided to go ahead and publish it for historic reasons this year in this journal with Kristen Hagees, who's at Cell Genesis, now at Celgene. And it was a CAR called CC49 Zeta, so first generation CARs. I said they didn't work in solid tumors. Here you'll see why. So this is an antibody targeting an antigen called TAG72 that Jeff Sloan at the NCI made too. And it was tested at Stanford and UCSF in some trials in the late 90s with this first generation CAR expressed with a linear vector. And we gave 3 infusions of 10 to the 10th cells. And, cells engrafted, proliferated. Now, I was showing you CAR T cells where we cure leukemia patients. It's up in this range. So we got about 30-fold lower than that. And then they went down to undetectable within 3 or 4 weeks. There was both an anti-idiotype response. There's a murine hybridoma responsible for that, and then probably the tumor microenvironment. We did have a very interesting thing done way ahead of its time, Indian labeling of a subset of the CAR cells. And so these patients had metastatic colorectal cancer, and the CAR T cells in some of the patients were infused directly into the hepatic artery, or in other cases intravenously. And this is, if you give just technetium-labeled albumin, this is the liver on this scan done back in the 90s. But if you labeled the CARs, this is what was seen. The liver was homogeneously labeled the CAR cells, except for where the tumor is, which is right here. So it was excluded. The TAR C cells did not get inside the tumor. And we didn't see any anti-tumor effects. We did see some, mild colitis, and this is the immunogenicity of the CAR, anti-EDA-type antibodies. So they didn't work. And solid tumors, we need specificity, and we need to be able to control checkpoints and a number of other issues, but I think they will. So one approach will be maybe targeting glycans, and this is work based off many years ago, started by Georg Springer, looking at the carbohydrates attached to serine and threonine-linked proteins, and where these sugar residues are attached with galactosyl transferases and an X-linked gene called cosmic, which is a chaperone for the transferase to work. And so it takes, it makes initially short sugars, and then they get progressively decorated, called different, core 1, core 2, et cetera, core 3 and 4, and usually terminate with sialic acid. Georg Springer recognized back then that this was an antigen then. If you had the loss of these sugars, that's something the immune system normally doesn't see, and that you could then make antibodies to the hypoglycosylated glycoproteins. And he predicted some really interesting issues back there in the 80s, that they're masked, and then they could be, these tumors do this, they could end up being targets. So then vaccine studies have tried to target TN in a variety of trials, but they did not target, it came out before the checkpoint therapies. So it's really been, abandoned. I think if it was looked at again now, we might see very different answers. So the TN, it's called T-Novel for the French, and SIL-TN is this. We also have validation in humans that it can cause immunopathology if you make antibodies to it. And both IgA nephropathy and the TN syndrome have been written about. And Rick Cummings in 2005 is the one who found that this cosmic gene, which is X-linked, when it's mutated, can lead to this hypoglycosylation. And this sometimes happens spontaneously in humans as an acquired somatic mutations, and then tumors do it either genetically or epigenetically. And importantly, this is pancreatic cancer. Almost all pancreatic cancers have this TN antigen expressed, and many solid tumors. Ovarian cancer is the same. So we don't know why solid tumors do this. They get some sort of presumably gain of function, and there's some data now using matched isotypic isogenic lines using CRISPR to knock out cosmic. It makes tumor cells happier to have that hypoglycosylation. But they also can now be a target with a CAR T cell against this antigen. And we're working with Henrik Clausen in Copenhagen, who's a carbohydrate chemist, to target these antigens. And with the idea that if you have MUC1 or mucin1 that's fully glycosylated, a CAR T cell against the hypoglycosylated antigen will ignore it. But if it has hypoglycosylated MUC1, then it will kill and be tumor specific. So we have an antibody called 5E5, which targets the hypoglycosylated epitope, and one called 5E10, which binds standard MUC1, which is expressed on normal colonic epithelial cells, and many cancers like colorectal cancer. And if you look at breast cancer, they both stain, and they have surface staining. But if you look at lactating or non-lactating breast, they expressed MUC1, but they don't express the TN antigen. And we're not able to see STN in healthy human tissues. Avery found that one of our favorite cell lines, the JERCAT T-cell leukemia line, has a mutation in cosmic. And that leads then to loss of function of cosmic, and then JERCAT cells express lots of TN antigen. Normal T cells do not, but the leukemic cell does, and they don't have the T synthase activity. And he showed both in vitro that the CAR T cell with 5E5 kills JERCAT very well. And if you do genetic complementation, if you add back in the cosmic gene, then the CAR T cells no longer kill a recomplemented JERCAT. So that's really specific, this antibody targeting a sugar domain, which is, I think, quite amazing. And this is what really is nice. So-called sweet CARs that target this sugar And this is a pancreatic cancer xenograft. This is an intraperitoneal tumor model with bioluminescence imaging. 3 weeks after CAR therapy, we get this with the sugar CAR, and with the CD19 control CAR, there's a progressive tumor. So quite exciting. So I'll just finish with this. And where we're all going to be as a field is combining these new therapies, checkpoint therapies, CAR T-cell therapies, with what we've had in the past. And it's not going to be trivial. There's many possibilities mathematically, and then how we do it is an issue. We have some very recent data that's exciting from Steve Schuster and Elise Chong of Penn on checkpoint therapies combined with CARs. So this is a patient who had at baseline a diffuse large B-cell lymphoma, 35-year-old guy who's multiply relapsed, end-stage DLBCL, invading his heart and pericardium. And we treated him with CART-19 here, and basically nothing happened. He needed ventilator support. So without any and this is what happened. The CAR T cells did expand in him, but to much lower levels than we see in CR patients. We gave him then a single infusion of Pembro, or an anti-PD-1. What happened was, there was a rapid increase in his CAR cells and reawakening of function. And within, this is day 45 here, the tumors began to look better. He's now in a CR, unmaintained. He was able to go from ICU care to a month later, he went back to work. So this is really exciting here. We now have a phase 1/2 study testing this. And this is data combining another targeted therapy. David Porter reviewed at ASCO a year ago the results in refractory relapsed CLL. And at the 3 centers, Memorial, Penn, and Seattle, the CR rates were between 25 and 45%. So not like we see in ALL. But when you do get a remission, they're very durable, and we haven't seen any relapses. We have an ongoing trial now where we're now taking patients who are on ibrutinib, which targets BTK, for CLL, and this puts people in a partial remission. They came into our study with having between 10 and 50% of the bone marrow still, full of CLL cells, but they're much reduced in their lymph node bulk compared to when they first presented. And then we treated them with a CAR cell, and they continued their ibrutinib, and we have had really lovely results. Not a single patient's required hospitalization for cytokine release syndrome, and 8 out of 10 have deep responses in the bone marrow, which with a CAR alone correlates with long-term disease-free survival. So we're very excited that both, combining a targeted agent like this will make it an outpatient therapy. None of our patients require hospitalization, and it may be then a curative therapy rather than what ibrutinib is, which is $180 a pill for the rest of the patient's life right now. It's much better than the alternative, which is they all die. But we need curative therapies with these expensive therapies, and I'm hoping a combination of targeted therapies plus cell therapies may get us there. So we're at this issue of moving healthcare from these pillars of pharmaceutical biotechnology and medical devices now to delivering ultra-personalized therapies. And it's a lot of complex issues. It's a new infrastructure. There are so-called N of 1 things where our only precedent for that really has been in transplantation and things like red blood cell transfusion. And how do we implement this now? I mean, initially there's going to be central manufacturing, but eventually I think this will be done at our local blood banks with machines that just manufacture these cells. And it'll be just a new form of transfusion medicine. The last thing I'll mention is the geographic disparity that's emerging with this kind of therapy involving gene transfer. So when we began in 2010, there were only 3 centers doing this, and you can enter the search term CAR T cells into clinicaltrials.gov, and as of last month, there are over 200 trials, compared to where there were here, and you can see the rate of rise. And you can geographically find out where these CAR T cells are, and there's 2 hotspots. They're the US and China. And in fact, in June of this year, China overtook us. It's quite amazing. They're investing more than we are in medical research. And it's geographically disparate, not just because of investment in dollars. You can look, to Japan, which has huge investment in biomedical research. They only have 3 trials open. So it's a society-based difference on how welcoming they are to gene transfer therapy and new therapies versus conservatism and many other issues. It's a very interesting thing to discuss. So, and unfortunately, there's nothing in the southern hemisphere. So we have big disparities, as we do with many other oncology therapies in that respect. The main issue going forward now is genetic enhancement of the cells and then manufacturing them in an automated way. And there's now an evolving group of engineers working on how to do this. We're part of a consortium with Chris Roy, who's at Georgia Tech, and Emory on how to do this. We're the only field right now that doesn't have standards. There's no NIST, for instance, for how to count cells, and we need that. And so synthetic biology is evolving, and it's requiring many different kinds of disciplines and so on. And it's a great opportunity, I think, for new careers in science. So this is just to sum up what I've said. I mean, our major scientific challenge now is solid tumors and how to make activity work there, and I think it's going to be complex. Interestingly, I didn't get time. We have seen activity in glioblastoma. There was a wonderful case report from City of Hope earlier this year on a single patient who had a really nice response to glioblastoma with an IL-13 receptor alpha-2 CAR, and we've seen similar with EGFR V3 CAR, but there's a lot more to be done in solid tumors. And TIL cell infusions work in these solid tumors. So how we'll get CAR T cells in depends on these issues I've discussed, and then manufacturing requires lots of work. So I've mentioned some of the people. Bruce Levine did all our manufacturing. David Porter took care of the adult leukemia patients, Stephen Grupp and colleagues at Children's Hospital, the pediatric patients. And we have a number of collaborators up in Cambridge and Novartis who've really been a great team to work with. So I'll thank you for your attention.
Carl, thank you very much. That was really spectacular. We have time for a few questions, but maybe I can ask the first. So how do we go from N of 1 to a universal CARs? And the question has been, using CRISPR today, removing MHC antigens, do you think we can get there?
So, I think in the end, Steve, we're going to have 2 kinds of therapies. One will be sort of knockdown therapies, induction. Those will be off-the-shelf, third-party cells, maybe coming initially from cord blood cells, later iPSCs, and so on. It's hard to engineer in what a T cell does, long-term selection and not be immunogenic. But there are a number of approaches, including here in Manhattan, people trying to do that with TCR knockdown, MHC knockout. And great data in very immunosuppressed children in London, these London baby cases where they made them resistant to Campath and were able to get cells to survive for 2 months in patients when they're third party and have anti-leukemic effects, but then remove the cells. So that's an ongoing line of research, and I think they'll complement each other.
Thanks. Ramon?
Yeah. My question's about the future for solid tumor treatment, and we know that a lot of solid tumors have a variety of signaling pathways that are shared with T cells that are activated, and how have you been thinking about strategies for trying to keep the T cell active and engaged biologically and target the tumor at the same time?
Yeah, so that's a huge problem, that, and then, the genetic complexity of solid tumors that's more than many other heme tumors. I think, so one issue, for instance, MEK inhibitors, for instance, can have effects on tumors and then on the immune system, and in some cases, they can be synergistic with, say, checkpoint therapies. We don't know yet with T-cell therapies. I think the advantage of tumor, because many, in some ways, activated T cells are very much like a tumor cell. I mean, they use a lot of glycolysis, and all those. So the advantage, I think, or the opportunity is using synthetic biology to in vitro hack the T cell in a way, and then, whereas if you would treat the whole body, you would then end up favoring the tumor cell and the T cell. So I think there will be a wider therapeutic index that way. But otherwise, there will be problems of, because of the shared pathways of very active RAS, for instance, in both T cells and tumor cells.
Okay, one last question, Julio?
Thanks.
Have you any evidence MRD in the patients that are, in these leukemia patients that are treated? And has there been any characterization of the MRD at a molecular level to understand why is it not being targeted perhaps by the CAR T cells?
So we've done MRD by NGS in CLL and ALL, and where we have a lot less data in diffuse large B-cell lymphoma, where unfortunately we don't get nodal masses, usually biopsy. But in ALL and CLL, these appear to be sterile remissions. We can't, down to level of 1 in a million, the leukemic clone is gone. And the kinetics for that to happen in ALL takes about 6 months. And it's an exponential decline. We don't really know. In diffuse large B-cell lymphoma, it seems to take longer. And so there's a lot left to learn on that. But the implication from what we're seeing right now in ALL is it may be possible to erase, if you will, or a suicide system, kill the CAR T cells if it's truly a sterile remission. And that's a kind of, bone marrow transplantation, allogeneic transplant, I see Jamie Farrar out there, the reason it works is you have long-term immunosurveillance. There's a lot of data on that. And so whether you're preventing the original tumor from coming back or another one because a patient has a genetic predisposition are all really important issues. I think what we'll have with CAR T cells eventually are the cells CARs turn on and off. And so you treat the patient long enough with an area under the curve where the tumor is no longer detectable, and that'll be done hand in hand with tumor modeling, and then and PK and so on. And then turn the CAR off, B cells immediately come back then. The B cell aplasia goes away within weeks when the CAR T cells either get rejected or turned off. But then if they could be turned back on again if they're needed, would maybe be ideal for this immunosurveillance. Because the long-term issue is going to be, patients without, if they're adults, it turns out the B cell immunodeficiency is really not a major issue because they have long-lived plasma cells that are CD19 negative. And, and you get those through vaccines and all your childhood experiences. Those hang around. And we have evolving data on that. The kids have never had all the vaccinations, so they have a much more severe immunodeficiency, and they don't get any long-lived plasma cells unless the CAR T cells go away. So they all require IVIG or gamma globulin replacement, which is a problem because that's a limited reagent, because it comes from blood banks. And, we can't have, so that's going to be an inexhaustible issue, just like the amount of oil under the sea. We're going to come to not have enough gamma globulin unless we have ways to reverse B cell aplasia.